fine wherever you are. I'm your teacher Ibrahim. I think you can still remember. In the fourth lesson, we are going to study how to estimate the cell size. I think you still remember that in our third lesson, we were able to make some onion epidermal cells, which I'm going to use in the fourth lesson to try and see how we can be able to estimate the cell size. There we go. It is right here on the screen. And remember, during that time when we are calculating, we are estimating the cell size rather, we need to know the diameter of field of view. Therefore, I need to put a ruler on top of the stage. Then upon putting the ruler, I can now come and try to see the measurements to know the diameter of field of view. Let me just try to look at it so that you can also be able to view it together with me. Diameter of field of view here, as I can see. There we go, there we go, there we go, we're almost there. Good. My diameter of field of view according to this is 2 millimeters. I hope you'll be able to see that. Let me just look at it keenly so as to be sure of what I've just seen. It's almost visible. It's almost visible. Good. There you go. You can just look at it. And before you look at it, I want you to look at what I'm going to write here. You will be able to see it looking like this. Remember, you will be able to zoom in and see the calibrations which are found on the ruler as some two dark structures. Those are the calibration. Then from here to here, that marks 1 unit, and unit in this case is millimeters. So you're going to look at the one that I've just seen, and you will be able to realize that it is 1 millimeter. Let me just try to zoom in and see. Let me just try to zoom in. Let me just zoom in. Diameter of field of view. Let me just take it easy, take it easy. Hope you have seen some dark structures alongside one another. Then that is my field of view, which are 2 millimeters. If you are able to see it very keenly, you would have seen something that looks like this. Then those are 2 units from here to here, that is 1 millimeter. Then another millimeter, that makes it 2 millimeters. So we have diameter of field of view as 2 millimeters. Field of view is equals to 2 millimeters. I hope you have been able to see there. My diameter of field of view is 2 millimeters. We get back to the microscope again and see the number of cells which are found along the diameter of field of view. There we go. I'm going to put this one here now. Since I've already known the diameter of field of view, I can now look at these cells of mine and be able to estimate their sizes. There we go. Let me just put it in the coarse adjustment knob to make it quite clear. When my images are clear, I'll let you know. Let me just try to tilt it to make them look. Let me just tilt it to make them appear clear. Perfect. You can now have a look at them. You'll be able to see some 4 cells which are closely aligned to one another. You will be able to see the 4 cells. Let me just view it slowly by slowly. There we go. There we go. There we go. Those are the cells that you can be able to see. I hope you are able to count them. There are 4 cells in my field of view. We can go to the next step, whereby we are going to look at the formula that we are using. During the cell estimation process, we use this formula over here. We say that the cell diameter is equals to diameter of field of view divided by the number of cells. My diameter, I've said, cell size is equals to diameter of field of view, which I've already written here. Let me just write it here, which is 2 millimeters, divided by the number of cells across the field of view. I had 4 cells in my field of view. So diameter of field of view needs to be converted into micrometers, because most cells are very small, hence you can't measure them in terms of millimeters. Therefore, we need to convert the measurements, the readings of the diameter of field of view from millimeters to micrometers. 1 millimeter is equivalent to 1,000 micrometers. Therefore, we are just going to multiply these 2 millimeters by 1,000 so that we can have the micrometers. Therefore, I'll come and say that cell size is equals to 2,000 micrometers divided by, let me just write the units of micrometers, that's how it's written, divided by the number of cells. If we divide this, we'll be able to obtain the cell size. In this case, the cell size will be, if you divide this 4 here, once 4 into 2,500. Therefore, the cell size is 500 micrometers. That is how we estimate the cell size. Therefore, that marks the end of our 4th lesson under the microscope, and I would request you to stay in touch. Please ensure that you watch my next video that is about the differences between light microscope and an electron microscope. Thank you so much for viewing this video. Please remember to like and subscribe. We'll meet in our next lesson when I'll be talking about